Okay, so this is the introduction to cells. We have a picture here that shows an animal cell and a plant cell. You're going to be responsible for knowing the differences between these particular cell types and some of the different organelles. You're also going to need to know the function of the different organelles. I will point out on this particular slide that right here we're missing a label for the endoplasmic reticulum. This is found in animal cells, and you'll see endoplasmic reticulum is labeled in plant cells. Many of these organelles are repetitive. They're found in both animals, plants. They're even found in fungus and protozoa and other organism types as well. So we're going to start out, though, our study identifying some scientists. Now, there are a bunch of cell scientists on here. Cells, being the basic building block of life, were not always what we thought life was based upon. But it was the investigation and the study of living things done by Robert Hooke, who identified box-like structures using his own homemade microscope that he was observing cork. So he took slices of cork, which are dried up plant cells, and he was able to coin the term cell. Anton van Leeuwenhoek saw single-celled organisms under microscopes that he made himself. Schleiden and Schwann were able to definitively determine that plants and animals are both made of cells. And Rudolf Virchow was able to determine that all cells come from pre-existing cells, which is a fact that has become part of what we call the cell theory, shown below. And Robert Brown determined that all cells, not all cells have a nucleus. He discovered the nucleus in cells. Now, there are a couple scientists that are more modern that are not included here, and I think I would be remiss to not add them. So I'm going to take you over here. Here's our Robert Hooke. All right, this was the year here is about 1665, so quite a long time ago. And we have Anton van Leeuwenhoek. That's 1674. He built his own microscope. That looks more like a medieval kind of torture device, but that is his microscope. It had a single lens, and he was able to look at pond water and identify single-celled organisms. Types of things that he observed: blood, sperm, scrapings from his own teeth. So there were probably bacteria in there that maybe he was observing. I'm not sure, but scrapings from your teeth sounds pretty gross to me. Henry du Rocher, we jump ahead about 150 years or so, and we get Henry du Rocher proposing that all living things are made of cells. We have Robert Brown, identifies the nucleus, and Schleiden, Schwann, and those are still 1800s, and Rudolf Virchow. Now, that covers all the old dead guys because they're all old, they're all dead, and they're all guys. But more modern science is being done by females than ever was done before, and it's important to recognize that Janet Plow in 1931 was able to identify the actual structure of cell membranes, which we identified earlier in our course as being a phospholipid bilayer. And she recognized, using her own investigation, that there's proteins and all sorts of carbohydrate chains lumped into this, and it's fluid, and it can shift and bend and fold, and it's very functionally useful for cells. We're going to see a lot of different organelles that are made up of cell membranes. Next, we have Lynn Margulis, and Lynn Margulis was in 1970. and she was able to determine that uh, mitochondria and chloroplasts are formerly free-living organisms that have their own DNA and are able to replicate on their own. So a very important contribution to our scientific investigations. I'm going to include here, real briefly, a graphic that shows a timeline of these uh, investigations and contributions, just because I put it together and I feel like it's a little helpful. So now I'm going to jump back to our previous. This is not in your note sheet. So if you're watching this video, you're probably getting a little bit of benefit out of this that you wouldn't get from just having your regular notes. So here I am back at your regular notes. The cell theory has three parts. It says all organisms are made up of one or more cells. So you have single-celled organisms and you have multicellular organisms. All cells have to carry on the life activities we learned about earlier in the school year. You should be familiar with some of those. And new cells only come from other pre-existing cells. Now, there are some problems with that idea. It's kind of like the chicken or the egg issue. What came first? So where did the first cell come from? And this is a problem for science, but we readily admit that that is one of the flaws in the cell theory, and we don't have an answer to how the first cell came about, but we do have some hypotheses. Here are the two types of cells that are found on the planet. There are prokaryotic cells, which are bacteria, and they don't have complex organelles. And then you have eukaryotic cells, which do have a nucleus, and they'll have lots of other organelles as well. All right, so now we're going to spend a little bit of time going over the organelles. I will tell you this. It would be very helpful, and I'm strongly recommending that you create flashcards for your scientists and that you create flashcards for your cell organelles. It is a major part of what this particular unit is about. So the cell membrane is the outer boundary that allows certain materials to move in and out. It is what we call selectively permeable. Some things can go through and other things cannot. Usually it's things like ions can't get through without having a protein channel. These little pink items in the little pink proteins embedded in here are channels to allow large molecules or ions to go through, but water can pass through it very easily. Right? Water can diffuse through cell membranes. All right, the nucleus. It's the control center of the cell. It has some important parts. It'll have the chromatin, which is DNA that is bound up with proteins. I have another alternate image that I'm going to show you for that. It has a nucleolus, and it can actually have multiple nucleoli if it needs them. This is where ribosomes and RNA is going to be made. So it is the copying place for the DNA. The nuclear envelope, the entire nucleus is wrapped in a membrane. So it is important to recognize that many of our organelles are enveloped in membranes. And I can show you that. Oh, that's the prokaryote and eukaryote stuff. Where's my nucleus? Here it is. Cell membrane, cell wall. Oh boy, come on. I thought I was going to click that nucleus right off the bat. Of course, it's never the one that I wanted. Oh, is this it? This is it. Okay, so this shows chromatin. The nucleus actually has nuclear pores that allow RNA to exit, but DNA stays inside. This shows the DNA is actually wrapped in all these little blue and white proteins in here are designed to help coil up the DNA, and it allows chromosomes to be formed. This is a picture of a chromosome, It is an original. One half of this X is an original. The other half is a copy. And the original and the copy are held together at what's known as the centromere, right? A chromosome is only going to be visible when the cell is actually divided. Okay, I'm going to go back to our note sheet and we'll head on to the next set of organelles. Cytoplasm is the watery material inside of the cell. It's where many of the chemical reactions occur in the cell. Many chemical reactions occur there. You have membrane-bound endoplasmic reticulum. So this is a series of folded membranes and channels, kind of behaving, I would say, like a subway system or a transit system to move materials around the cell. So this is what it looks like in real life using an electron microscope. This is what you'll see in the cartoon type diagrams. And there are two types of endoplasmic reticulum. You'll notice that it actually joins right up with the nucleus. And there are some endoplasmic reticulum that has red dots. Those dots represent ribosomes. We call that rough endoplasmic reticulum. And then there's also smooth endoplasmic reticulum that does not have ribosomes on it. What are ribosomes? Ribosomes are the site of protein synthesis. They can be free-floating in the cytoplasm, or they can be attached to endoplasmic reticulum. Now, a memory clue would be to think of ribosomes like ribs, like that's meat, all right? So ribosomes, or ribosomes, depending on who you talk to, are going to be making proteins, and meat is protein. They're also made out of RNA. This is rRNA, which is ribosomal RNA. So they're genetic material as well. Golgi bodies are membrane bound. We have a nice picture here that shows the electron microscope image in real life is in black and white, but then you have the cartoon version here. The Golgi body is like UPS. It's responsible for packaging and secreting or moving materials out of the cell. A lot of different cell proteins and secretions are finished there, and then they're sent out for the rest of the organism to use. Vacuoles are a storage area. Plants have a large central vacuole that helps to keep them rigid. We use the term turgid. It helps it stand up. All right, and so when plants get dehydrated, this central vacuole kind of shrinks and the plant wilts, falls over under its own weight. It's a great storage place for food, water, or wastes. Plants and animals have vacuoles, and they come in different varieties. Here's an example of a different type of vacuole. A lysosome is a vacuole that has enzymes in it to digest worn out cell parts. That root, lyse, you've recognized it, hopefully. A lysosome, like the term hydrolysis, lyse means to break down. And lysosomes will fuse with old, worn out cells and cell parts, and it'll break them down. Our cells also use lysosomes to help kill bacteria. Mitochondria. The mitochondria, I know that in middle school you learned the mighty mitochondria, or it's the powerhouse of the cell. I'm going to challenge you to change that thinking a little bit. I want you to think of the mitochondria as the site of cellular respiration. We need to up our vocabulary here. Cellular respiration is a chemical process where sugar is converted into ATP. ATP is energy for our cells. So you eat sugars and you get energy from those sugars. But the sugars have to be taken to the mitochondria where they combine with oxygen to make ATP. And the waste product in this case is carbon dioxide and water. right? The mitochondria is a double membrane. This is where the cell scientist Lynn Margulis was fantastic in identifying that these could change and modify and duplicate themselves on their own because they have their own DNA. But you'll see in here there are two sets of membranes. There's an outer membrane and an inner membrane that's folded. And I want you to think, if you were to take that inner folded membrane and stretch it all out, it would be much, much larger than the actual mitochondria, because those folds increase the surface area inside of that mitochondria, meaning they can pack in more proteins and enzymes to do the job that the mitochondria needs to do. So that those folds increase surface area to make reactions happen much faster. Those folds are labeled here. They're called cristae or crista, depending. There's oftentimes there'll be an E at the end when it's plural. All right, so in plant cells, which also have mitochondria, there are some organelles that are only found in plants. One of those is the cell wall. Cell walls are rigid outer skeletons to provide support. They're typically made out of carbohydrates, cellulose. Right? When you look at a plant cell under a microscope, they all kind of look uniformly shaped. And that's because those cells are surrounded by walls that keep them pretty rigid. In addition to having cell walls to support their shape, they also have chloroplasts. And these are the photosynthetic organelles in the organism. Light energy is changed into chemical energy, converting carbon dioxide and water into sugars. This is why plants need water, plants need carbon dioxide, and then they can make their own sugars from that. Right? That's what photosynthesis is, and it's done in the chloroplasts. The chloroplasts have a double membrane also. You have a membrane on the outside, and then you have all these folded pancake-like stacks, or they look like stacks of coins. And if you think of them as stacks of coins, you might think it's money. It's energy currency for the plant cell. So these stacks of coins are called thylakoid membranes, and those are grouped into stacks called grana. Only found in animal cells is the centriole. Centriole is the bundles of microtubules, which are part of the cytoskeleton. These are packaged pretty uniquely. They're in 9 groups of 3. So they make this kind of flower pattern, or some people have described it like it looks like a hurricane. Other people have described it as looking like Twizzlers. So these are the kinds of things that they get described as. They come in pairs. There are only 2 of them, and these 2 are located at what is an area known as the centrosome, and they help with cell division in animal cells. Plant cells do not have these, but they still do cell division just like animal cells do. They just don't have centrioles. All right, so cells are just the functional building block. Cells that are similar, that do the same job, can be grouped together into what we call tissues. Right, so you have muscle tissue, you have nerve tissue, you can have connective tissue. There's fat, which is a tissue. All right, so all these different types of tissues, they do the same thing. Now, groups of tissues that work together to do a job are grouped into what we call organs. So your heart is an organ. It's made up of muscle tissue, blood, which is another type of tissue. It has connective tissue, all these different types of tissues working together to do a job. The heart is also grouped with other organs to complete a whole task. So the circulatory system, right, or the digestive system, or the reproductive system. They're all systems of organs that are working together to do a job. And lastly, beyond the different organ systems that you have, is the organism. You can't remove any organ system and have the organism live. So these are in order of greater complexity from top to bottom in this chart. All right, so I'm going to stop right there for today, and we'll look at the movement of materials across the cell membrane in our next video. Have a great day.